Our laboratory in the Medical Research Council Protein Phosphorylation and Ubiquitylation Unit, or MRC PPU for short, studies how cells recycle mitochondria by a process called mitophagy. Mitochondria, the cell's power stations, must be recycled when they get old. A failure to do this in the nervous system, specifically neurons in the brain, can lead to neurodegenerative diseases such as Parkinson's disease. We have developed a stain, shown here in this video, of live human cells, which allows us to measure mitophagy. Healthy mitochondria have greenish yellow colour, while those being recycled are red. For scale, the healthy mitochondria appear as worm-like structures and are approximately 5 millionths of a metre long. The recycling mitochondria in red are much smaller as they have been broken up into smaller pieces for recycling. We think that boosting mitophagy in the neurons of people with Parkinson's disease may be a way to treat the disease. To see if this is possible, we need to be able to watch this process happening in these neurons. We use mouse brains that have our special mitochondrial stain to study this. First, we need to make very thin sections of the brain using a sledge microtome, which is somewhat like a mandolin slicer used to cut food. The brain needs to be very cold to make thin sections, so we place the sample on a stage cooled with dry ice at -78.5 degrees Celsius and fix it in place with a sugar solution. Once the brain is frozen, we cut 50 micron thick sections, which is about the thickness of a human hair. We place these sections in a plate containing PBS, a physiological salt solution, and stain them, which takes a series of steps. We remove the PBS from the plate, add a detergent and place on a rocker. Small holes are made in the brain sections by the detergent. After 15 minutes, we remove the detergent and add an antibody. The antibody can get into the brain tissue through the small holes and mark the dopaminergic neurons. It is these neurons that die in Parkinson's disease. We place the plate on the rocker overnight. The next day, we remove the antibody and add a second, which includes a blue dye that will let us see these neurons under the microscope. We place the plate on the rocker for 1 hour so that the second antibody can stain the first. Finally, we mount the brain tissue on microscope slides. To do this, we select a piece of tissue, rinse it in PBS and place it on a slide. Mounting liquid is added, a cover slip is placed on top, and its edges are sealed with clear nail polish. We can then go to our special high-resolution microscope to take pictures. After loading the slides, we locate the area of interest and take a picture. Here we can see mitophagy occurring in these specific neurons. We can also reconstruct the images in 3 dimensions to visualize the areas where mitophagy is happening. This research is allowing us to determine when and where mitophagy goes wrong in diseases such as Parkinson's. This will allow us to find potential treatments that can restore or improve mitophagy and hopefully aid in finding a cure for this debilitating disease.